Right, so next is the Golgi apparatus. The Golgi apparatus, or sometimes just called the Golgi, people leave off the apparatus. And so is a stack of 3 to 10 membranous pouches. Again, these are called cisterns or cisternae. Okay? And it looks kind of like a stack of pancakes. Golgi apparatus is going to look different from ER because usually Golgi apparatus is kind of more tightly stacked, whereas the endoplasmic reticulum really can be all over the cell. It doesn't have to be like limited to this area. And usually the pouches are like really long or really wide, whereas the Golgi is in pretty tight stacks, so it really does kind of look like a stack of pancakes. Right? So basically what happens is endoplasmic reticulum produces, these proteins or these carbohydrates or lipids, and it packages them in little spheres called vesicles. And those vesicles carry whatever the endoplasmic reticulum produces to the Golgi apparatus. What the Golgi apparatus is going to do is it's going to sort and then transport those molecules to the correct location. So in a lot of ways, the Golgi apparatus is kind of like the post office of the cell. It receives the package from the endoplasmic reticulum, and then it reads the address and says, oh, you're supposed to go to the membrane, or oh, you're supposed to go to some organelle inside the cell. So the Golgi sorts and then also uses vesicles to transport those substances to the right location. Now, one of the things that's interesting about the Golgi apparatus, I know sort of this, what we're learning here seems like really, really detailed stuff. Like, when am I ever going to use this? And technically, in medicine, you're going to use this every day. Understanding disease happens at the cellular and molecular level. And so if you don't understand your cellular and molecular level very well, you're not going to understand disease very well or how to treat it. So one example that I like to give when we talk about this is a very specific disease that affects the Golgi apparatus. This is actually a really awful disease called Tay-Sachs disease. You'll see it in your notes. I didn't put it in the slide, but you'll see it in your notes. This is a lethal genetic disease caused by a malfunction of the Golgi in the nervous system, in the brain, in brain cells called neurons. So these neurons, the Golgi malfunctions, and so the neurons produce these proteins, but the Golgi doesn't know how to sort them and send them to the right place, and so it doesn't. It just stores them. That doesn't mean that means that the ER is still going to be producing this stuff, but the Golgi is not doing anything with it. So the Golgi just fills with this backed up protein and eventually squeezes out everything else and kills the cell. And it's actually really devastating because this disease really takes its course very quickly. It usually causes death in infancy, usually within a few weeks to a month. So it's a really awful, devastating disease. But it affects this very specific organelle. And next is the mitochondria. So the singular form of this is mitochondrion. So don't be surprised if you see mitochondrion in certain situations. So it would be R-I-O-N instead of R-I-A. Okay. Mitochondria is plural. We're talking about more than one. Okay? So mitochondria are tubular spaces that are like the nucleus surrounded by two phospholipid bilayers. Okay, so notice here that when I look at this mitochondria here, there's kind of an outer darker membrane and then an inner lighter, like more orange membrane. And the inner membrane is much more folded. The outer membrane isn't so folded. Okay, so those are two separate lipid bilayers, two separate membranes. Okay, so the outer one is called the outer mitochondrial membrane. The inner one is called the inner mitochondrial membrane. And like I mentioned, the inner one is highly folded. Those folds of the inner mitochondrial membrane are called cristae. Now, inside the inner mitochondrial membrane, you're going to have a fluid-filled space that's called the matrix, or the mitochondrial matrix. And the mitochondria inside this matrix has its own DNA. The DNA in the mitochondria is circular. That's why they show you kind of a purple circle for it. So the chromosome is actually circular. It's not like linear. like our other chromosomes in our nucleus. And it also has its own ribosomes in the matrix, which is really weird, right? Like this mitochondria has its own DNA and its own ribosomes. Kind of seems like it could be almost its own cell, right? And a lot of people think that at some point in evolutionary history they were. So if you're interested in this kind of thing, look up what's called the endosymbiotic theory, where basically people think that eukaryotic cells derived from prokaryotic cells that were ingested, and then the two cells, instead of trying to fight each other, just became partners. So this looks a lot and actually behaves a lot like a bacterial cell. Okay? Now, what's the basic function of these mitochondria? So the basic function of the mitochondria is to produce most of the ATP for the cell. Now, what is ATP? ATP is basically a chemical form of energy that will be used by the cells, like the cell's money, right? And inside cells, nothing's free. Just like in real life, nothing's free, right? You got all these organelles and proteins that are doing things inside your cell, and they're not going to do it for free. You got to pay them. And it turns out you don't pay them in money. You pay them in this chemical form of energy called ATP. Okay? And so the mitochondria is what produces most of that. Last of our membranous organelles are vesicles, lysosomes, and peroxisomes. Now, why do I lump these three together? Because technically, structurally, they're the same thing. The only thing that will be different is like what's inside of them. So vesicles, lysosomes, and peroxisomes are all fundamentally really just a spherical organelle. Sort of like this. Kind of imagine a tennis ball and cutting a tennis ball in half. So inside the vesicles, lysosomes, and peroxisomes, you got this space, and then the wall of it forms a sphere that's made of phospholipid bilayer. Okay, so vesicles, the difference between the three of them is basically what's inside of them. Structurally, they're the same things, just they have different things inside. So vesicles, much more variable. Vesicles are kind of like little train cars inside the cell that can carry all kinds of things from one place to another. So what's inside a vesicle is really variable. But lysosomes and peroxisomes are going to have very specific things inside of them. Lysosomes very specifically will have digestive enzymes inside of them. Okay? So they're small, like you'll see the descriptions here. They're small membranous spheres containing digestive enzymes that are used to digest substances like cells or molecules in those vesicles that fuse with the lysosome. Okay? So lysosomes kind of like for digestion inside the cell. Peroxisomes are small membranous spheres that contain enzymes called oxidase, catalase, and other enzymes. And what these enzymes do is they detoxify substances produced in the cell, like free radicals and hydrogen peroxide. All right, so let's start a quick question here. Normal diploid, right? Diploid meaning 2 copies of each chromosome. Human cells contain blank total chromosomes in the nucleus. Right? So 2 copies of each. Right? So remember the correct answer for this one is B, 46. Right? So you should have 23, but diploid means you should have 23 pairs. So 2 times 23 is 46. Okay, 23 would be the haploid number, not the diploid number. Okay, this would be what would be in gametes, not in normal somatic cells. Okay, remember 92 is definitely not what we're going to try to go for. 47, 48. Sometimes people have these chromosome numbers abnormally, but never normally. Okay? Next question. Which of the following organelles produces secretory proteins? Secretory proteins like insulin. I try to give you guys a protein you probably heard of, right? Secretory proteins. Okay, who produces that? Okay, so the correct answer to this one is D, rough ER. Rough ER is what produces the secretory proteins. Remember, smooth ER produces carbohydrates and lipids.